First of all, happy Swine Day. I call it in the Animal Science Department when I'm visiting with all the support staff. I say this is National Pig Day, but the cattle guys get upset, so I need to, when I'm here, be professional, right, Dr. Odie, and call it KSU Swine Day. Want to welcome everyone. We're glad you took time out of your busy schedules. The whole morning we've been rolling all the sponsors for the different events throughout the day, and it was exciting last night. We had a reception, the students there, a lot of former students as well as supporters. Gary Fitzner came in and he said, right away, he goes, well, I want you to know I came the furthest to the reception last night. And I said, okay, where'd you come from? And he goes, San Diego. And then Dr. Fu from China walked in and gave me his Chinese card, so I gave it to Fitzner this morning to make the point. With that, we're going to have a very busy day. It's going to be a fun day for us in terms of sharing research results and extension activities. The program, many of you have seen it already, but the entire program except for lunch is going to be in here. Lunch will be next door. Usually Dr. Nichols and Dr. Odie, my boss, just asked me why Dave's not emceeing today. Dave's out recruiting. He should be back from Louisville. He's recruiting students, undergraduate students at Louisville, Kentucky. And last year I introduced him as the MC, as the MVP of our department. Well, actually, he went out and broke a new record, and I'm sure Dr. Odie's going to share this. We have 1,250 undergraduate students in animal science at K-State. And so Dave and his teaching faculty has just done a remarkable job. And not only do they recruit a lot of students, but they also do a tremendous job advising. The person that coordinates and oversees all this is Dr. Odie. He's our department head, and he's going to give us our official welcome to Swine Day this year. Dr. Odie?
Thanks, Jim.
Thank you very much.
Good morning and welcome to Swine Day. This is one of my favorite days. We're very, very proud of the swine team at Kansas State University, and they do an outstanding job of putting this event together. I would like to take a moment today and just maybe share a few thoughts with you that are perhaps a little different than what I've done in previous years. And I want to talk just a little bit about a few things going on at Kansas State University, and then a bit more about some things going on in the Department of Animal Sciences, and perhaps some of the factors that are influencing how we think about how we both educate students and how we conduct research and conduct extension programs. We have at Kansas State University right now a presidential initiative that we call the Global Food Systems Initiative. And we think this is a really big deal to get a institutional focus around food and agriculture. This obviously creates a lot of opportunity for the College of Agriculture. I think it will create a lot of opportunity for our faculty in the Department of Animal Sciences and Industry, and we're really pleased that the institution has decided to elevate global food systems as a priority for the institution. In fact, if we look kind of underneath the Global Food Systems Initiative, we have long had a focus at Kansas State University in a variety of departments on not just agriculture, but international agriculture and actually global agriculture. We have in the last 2 years, these have been announced at Kansas State University. There are 4 Feed the Future Innovation Labs that are USAID funded. USAID is the United States Agency for International Development. And in total, these 4 big grants are over $100 million. So, and most of these are on the plant side, but we do have some animal agriculture incorporated into some of these areas. But this is a big deal, and it's a big deal for Kansas State University. And I think it really reflects the quality of people, the quality of faculty that we've had at Kansas State for a long, long time. These don't happen by the administrators of the university. These happen by the faculty of the university. It's the faculty that really lead these initiatives, that write the proposals, and are really successful in actually delivering these. And I think this is a rather big deal. We also live in what I think is a really interesting time as we look at a university and we look at businesses like you folks are engaged in. We are facing very, very rapid population growth. I'm sure many of you have seen this, but we're looking at out here by 2050 having around 9.5 billion people on this planet, and we're somewhere in the perhaps 7.5 billion people right now. And I contend that this is a really big deal, and it's a big deal for all of you in this room, and it's a big deal for all of us here at Kansas State University. If we look at the nature of that growth, we're going to see more in this older population. But what we're also going to see associated with this growth is not just more people, but rising standards of living in the developing part of the world. And associated with those rising standards of living, stronger economies, we're going to see more demand for food, and particularly more demand for foods of animal origin. And I think that's going to be very favorable for our industries. Food demand is forecasted to increase by 60% by 2050. And if you look at the world meat market today, this is the world meat market today, and you look at the sources, so this was 2014 forecast, 311 million tons. Bovine was 68 of that. Poultry was 108 of that. Pork is 115 of that, and ovine is 14 of that. So if you look at the global meat market, pork is the largest single meat consumed on the planet. And we're going to talk just a little bit about what we think that's going to do as we look into the future. If we look at what forecasted growth in global demand for meat is, and we look at it in the developing countries versus the developed is largely United States, Canada, Western Europe, Japan, Australia, New Zealand, and the developing parts of the country are most of the rest of the world. So we look at 2002 per capita consumption of meat, 28 kilograms. By 2050 in the developing countries, that's forecasted to go to 44 kilograms. In the developed countries, it's forecasted to go from 78 to 94. Generally, as standards of living increase, consumption of meat actually goes up. And if we look at total meat consumption in megatons, forecasted to go from 137 to 326 in developing countries and 102 to 126 in the developed countries. And this is expected to be disproportionately on the pork and poultry side. So I'm more of a cattle person. We do expect demand for beef to grow, but not at the same rate that we actually anticipate it growing for pork and poultry. So I think the pork industry has an enormous opportunity here in this changing dynamics that we're seeing globally. So what does the future look like? This is kind of my crystal ball. Growing demand for U.S. meat, especially pork and poultry. I think that's what we'll actually see. But not only that, and I think many of you that are in the business are going to be beneficiaries of this, but I think an increasing percentage of the dollar that producers receive that comes from the consumption of pork will come from export markets. I think an increasing percentage will come from export markets. I think if you look at this 60% growth in demand for food, we're going to see a growing demand for science and technology. And the U.S. is the leader in the development of science and technology. And I think this is very important for us. I think it's a great opportunity for us. But I also think it's a message we have to be delivering to our policymakers and leaders, and it's the importance of public funding, of investment in science and technology. And we've lagged in that area in the U.S. now for quite a number of years, and I really think it's an important message, and we need all of your help in trying to convey that message to our leaders as we look at both state and federal government. I serve on the board of an organization called the National Association for the Advancement of Animal Science. We actually have a lobbying group presence in Washington, D.C., and our strategy is really to try to grow a federal investment in agricultural research, and specifically in the area of animal agriculture research. We also think this will result in growing demand for well-educated, and I like to look at this as globally knowledgeable workforce. So as we think about educating students here today, I think it's more and more important that they be knowledgeable about the planet, and not just Kansas or not just the United States, but really have a a global picture because I think that's where a lot of the opportunity is actually going to be. So if I look at our priority, I don't think our priorities really change very much. It's high quality, hands-on education for undergraduate and graduate students. We have 1,250 undergraduates. We have about 240 graduate students. About 140 of those are distance food science students. We also, I think this is as important as it ever was, high quality, relevant. I really like, this is to me a very important word. I think a lot of the reason you folks are here today is because this swine team at Kansas State University, I would argue, has done relevance probably better than anyone else has actually done it. And I think that's a really big deal, that their engagement with you folks and the people in the swine industry really has led them to be a highly relevant organization. So high quality, relevant research and extension programs that serve the livestock industry. So with that, again, welcome to Swine Day, welcome to Kansas State University. We're delighted to have you here, and I will pass it back to you, Jim.
Dr. Odie gave us that presentation at a faculty meeting in October, and I asked him a couple weeks ago if he'd share some of those slides, because this message continues to be given by folks across the U.S. and around the world. And even our animal science meetings this summer, I usually don't notice the detail, but they put meeting the global demands for food in 2050 on the bags they handed out with all the relevant publications. Having said that, right after Dr. Odie gave that presentation in October, Science Magazine came out, and the president of Stanford University addressed the need for animal agricultural research dollars to meet that demand, and he really emphasized that the money today in Washington is going to the plant side. And so I think his message was clear that we need to meet that demand. Speaking of being relevant, we need to stay on time this morning. Last year, Dr. Henry and Dr. Hesse gave us an update on PED virus. We thought it was going to be a short-term situation. Matter of fact, I said at the talk last year, you know, some people said it was going to be an old disease by the time Swine Day came. Well, we're 1 year further into it now, Dr. Hesse, and he gave a presentation this year at the Lehman Conference on PED with his colleagues, Dr. Dreetz and Dr. Woodworth, that was well received. We asked him to come back and update us on what they've learned, not only on PED, but Delta coronavirus as well. Welcome back, Dr. Hesse. Let's give him a round of applause.
Thank you.
We look forward to it.
Okay, yep, very good. Okay, so, yeah, you can hear me. Okay, so last year Steve and I talked about PED. This year we have a new bug that I get to share with you. This bug is porcine Delta coronavirus, and you're kind of a special group because you're basically the second group of people that have heard this talk. The reason that it's a special talk is this is the first time in the world that Koch's postulates have been fulfilled with this agent. That is a big deal, by the way. Okay, so a colleague of mine, Ian Zan, from the Ohio Department of Agriculture, was actually the first one to start to track this virus. He started to see that back in probably November of last year, and they get wave infections following PED going through herds, and the virus that they pulled out, or the thing that they pulled out, did not react with the standard PED diagnostics. Something was different. And Ian and his group, along with researchers at Iowa State, but mainly out of Ohio, they started to do the EMs, find out it was a coronavirus. Then they started doing the molecular biology, getting some sequence back, and lo and behold, they found Delta coronavirus. That's a really unusual thing. They actually published this data in Emerging Infectious Diseases, and this is a publication. In September it came out, but it's been around for a while. Delta corona is, obviously it's a coronavirus, but it's the first time that Delta coronaviruses have ever been shown in pigs. The original description of them actually came from slaughterhouse samples out of, I think it was Hong Kong, and that's where the first description of them occurred. So this virus, once again, has an Asian signature to it. It's an interesting thing. I'll talk a little bit more about that today at the DMP seminar. And it's different than the alpha coronaviruses, that the TGEs, the PEDs, and then some of the beta coronaviruses, the HEVs, that we normally see, that we recognize in swine. So this is a totally new emerging virus, and we don't know really anything about it, except for what I'm going to share with you now. All right, it is an important virus. The USDA is constantly tracking it, just find out where it's at, where it's going. It's prevalent nicely. It's all over the United States. It continues to be detected. When we first saw it, we thought it was going to be a blip on the radar screen, come and go, but it continues to show up. And this is as of last week, I believe, yeah. As far as the states that have it, it's also been described in Canada, other countries in the US. Doesn't seem to be in Europe right now. They're really interested in what's going on, and they're following what's happening in the US. The disease. So, basically, it looks just like TGE. It looks just like PED. It's just not quite as bad, which is a good thing. Obviously, the place that it has the most effect is in the sow herd. And I'm using PED slides because they are so impactful. And too many people are belittling this disease. But in reality, if this was Delta Corona, that pickup truck would be half full instead of as full as it is. So this is a significant disease, and we need to be paying attention to it. Really hammers young nursing pigs. That's where you see the most effect. But in older pigs, it will also cause diarrhea, just like TGE, just like PED. And diarrhea is what you're going to see. It will infect sows, cause them to develop diarrhea, cause them to get sick and vomit. So clinically, you really can't distinguish between it or the other alpha coronaviruses that currently infect swine. Going to have to use good diagnostics. All right, so now that's a little background on PED. That's the historical background as it was described in the field. So now we're talking about the actual fulfillment of Koch's postulates. This is a grant that was funded by the National Pork Board. The goal of it is to determine the sites of tissue localization, routes of shedding, duration of virus carriage, kinetics of antibody response, and also whether or not it has the potential to generate aerosol infection. This is a team effort. I really want to emphasize team. Researchers at K-State, at UNL, is that showing up? Yeah. At UNL and at South Dakota combined resources and efforts and time and energy to actually pull this study off. A lot of the laboratory work was done right here at the Veterinary Diagnostic Lab at K-State. Actually, all of it was. The animal work was done primarily at UNL. They have excellent animal facilities that suit the needs for this type of study. We can actually work in farrowing crates. That's critical to the type of diseases that we're seeing in the swine industry. We don't have that capacity here at K-State. We need to consider how to get it. And then also Eric Nelson at South Dakota. He's a fellow diagnostician. He has access to viruses, strains, samples. We share and share alike among the diagnostic labs. That's how you get things done. And as I said before, the National Pork Board provided the funding for this. Experimental design, okay. So the study itself was done at UNL, as I said, BSL-2. We can actually do work with pregnant sows, freshly farrowed animal baby pigs, a good place to do it. The study itself involved 10 pregnant sows and their litters, experimental groups, inoculated, oronasal inoculated, contact controls within the litter. So 6 were inoculated per litter, 2 contact controls. That's estimating a low estimate of 8 animals per sow. The sows were actually inoculated as well at the same time that the baby pigs were. We also had aerosol control sows and their litters in the same room with just a curtain in between, so they had a common airspace. So we're trying to address aerosol transmission there, and then obviously we had negative controls too, that were just held to see, make sure that everything's clean. The challenge was provided by Eric Nelson in South Dakota. What it was is a diagnostic sample that he received into his laboratory. He had two different ones that he had that looked to be real clean, one from baby pigs, one from sows. They didn't seem to have any other infectious agents with them. We didn't know which was going to work, but we had rumors from the outside that people at Minnesota and Iowa State and Ohio had tried to do what we're doing and failed. And the inocula, I think, is the key ingredient. Of the two inocula that Eric had, we amplified it in 2-day-old pigs that were transitioned to a milk diet. Only one of the two different inocula actually amplified, and we were able to generate enough challenge stock to do this study. Since then, we've taken this challenge material from our day 1, 2, and 3 pigs, generated a large pool of this, and we are sharing that with researchers across the world, basically. It's already gone to USDA in Ames. They're doing some of the work there. So, I mean, a lot of benefits from this study. Nursing pigs and their dams. The baby pigs were inoculated at approximately 2 days of age, 5 mils per route, orally or intranasally. And then a 2-day-old pig squirting 5 mils up their nose is close to impossible, but we did it. The study itself is, since we don't know much about it, it's important to understand pathogenesis, and that's what this study is really geared towards. We're collecting baby pig samples early on, where we're actually going in and sacrificing pigs from the different litters and collecting tissues so we can see exactly where the virus is replicating, how it transitions to other tissues, follow the immune response. So this is basically the experimental design. The first 7 days really intense on post-mortem examination, collecting all the tissues. We looked at blood, nasal swabs, fecal swabs. That's just the experimental animals, how they were sacrificed from the different groups. Are we getting feedback here? I'm hearing echo. Okay. And then also, once the baby pigs got older, we collected oral fluid samples, because that's an important surveillance tool that we have available to us. Does it work or not? We followed the pigs all the way out to 42 days post-inoculation so we could monitor serological response as well. And then also how the pigs performed after infection. Tissues, we basically serial sectioned the pigs to look at everything, both histologically and then localization of virus via immunohistochemistry. So what did we find? After inoculation, the sows did develop a mild diarrhea, 2 to 3 days post-inoculation, 3 days I guess, and they were clinically normal by 3 days post-inoculation. Now these are the inoculated sows. The aerosol contact controls, they also developed diarrhea, but it was about a day to 2 days later. They were also clinically normal by 8 days post-inoculation. So the sow is an indicator of infection. They will get sick. It's a mild disease. They will develop diarrhea. We didn't see any vomiting with this, so that's probably a differentiator between PED, TGE, and Delta Corona. The controls stayed clean. That's always good. We like to see that. Now, the baby pig. So what do we see with them? At 1 day post-inoculation, or I guess this is 2 days post-inoculation, the first thing that we noticed is some of the baby pigs got sick and vomited. They're clearly not feeling well, but they were vomiting. They're also starting to develop profuse diarrhea, huddling, getting under the heat lamp. 3 days post-inoculation, they'll die of very severe dehydration. You open them up, and the intestines are thin, very dilated. I mean, it's classical viral enteritis. All right. So, clinical disease really didn't show up until 2 days post-inoculation. And there was no difference in the onset of disease between the animals that we inoculated versus the contact controls. So horizontal transmission did occur, and it occurred very rapidly. Morbidity was 100%. Mortality was variable. Some of the litters got hit really hard with virtually 100% mortality. Some of the litters, even though they got sick, the mortality was maybe only 20%, 25%. So we don't know exactly why. We're just making the observations and sharing right now. I think that's all I want to say there. The aerosol contact controls, they also developed severe diarrhea following what their dams had. And they, actually, some of those got hammered even worse than the inoculated animals. So we're kind of surprised about that. All surviving pigs, whether they were the aerosol group, the contact control group, or the inoculated group, they became clinically normal at about 12 days post-inoculation. The negative control stayed negative throughout. All right, so that's the clinical picture that we're seeing here. At 14 days post-inoculation, as they were sacrificing pigs for sample collection, Dr. Broderson went ahead and took a picture. This is a normal pig versus a pig that has experienced infection. So pretty dramatic difference as far as cause and effect of inoculation. It really does set them back. All right, interesting data. Fecal shedding. Okay, so just a little bit about this curve. All right, day 1 all the way down to day 42. Green is basically no shedding. The shades of green are the less amount of virus being shed as measured by real-time PCR. If it's red, brick red, that is not good. That's lots of virus being shed. The more intense the red is, the more the virus is being shed. So 1 day post-inoculation, we did not see any shedding in any of the animals. 2 days is when we started to see it, and you can't really see too much from here, but the cycle times, the virus load is in the teens, mid-teens to low teens. That's a lot of virus being shed. Tapers off, day 3, 4, 5, that's where we're at. And then basically by 14 days, we're seeing some fecal shedding, not a lot. By day 21, only 1 or 2 animals are showing it. So this clears in about 3 weeks. It's a little different than PED because with PED, we saw shedding all the way out to 42 days. So that's another interesting difference. Fecal shedding of the different groups, I'm not going to spend a lot of time. The negative stayed negative all the way through. Inoculated contact controls, virtually identical patterns. The aerosol controls, it was just delayed basically a day, but the amount of virus being shed, virtually identical. Nasal shedding, same type of pattern. The amount of virus being shed is not nearly as much, and one has to ask the question, is this truly nasal virus that's being shed, or is it fecal virus in the nares? It's probably fecal virus in the nares, and I'll get to that in a little bit. All right, so this is, if you're a pathologist or somebody who studies pathogenesis, this is a really cool slide. So let me explain it a little bit. All right, so this is PCR results of the different tissues. So we have blood, serum, trachea, turbinates, tonsils, all the way down over into here where you're getting into the GI tract. Stomach, descending colon, spiral colon, small intestine, and cecum. All right, so the first thing that I looked for is viremia. And lo and behold, we're seeing viremia at 1 day post-inoculation. That's a day before we're seeing it being shed. So somehow, and that's a difference with PED, these animals are becoming viremic a day sooner than what we would see in a PED system. So viremia is detectable at 1 day post-inoculation. we can track it out to about 4 days post-inoculation, then the viremia disappears. Okay, that's the difference with PED. The other thing that's really kind of interesting, we didn't get this data till later on, when we looked at samples in the small intestine, at 1 day post-inoculation, the small intestine is just absolutely loaded with virus, but they're not clinical at this point in time. The virus is eating up the cells within the small intestine, leaking through the lamina propria into circulation. Now, the thing that's really cool about this from a pathogenesis perspective is, here's the small intestine right here, but look at the descending colon. Not much virus there. Timing-wise, we nailed it. So, from a pathogenesis perspective, mechanism of transmission, you know, the timing, we just hit everything perfect there. And our pathologist and graduate student at UNL is in the process of writing all this up. Other interesting things that we saw with sample collection over time is look at the small intestine over time. This is day 14 at the top, day 42 at the bottom. And we have 2 sample types that are really giving a constant signal, and that's lymph nodes and small intestine. So the virus is being harbored in these tissues, or let's just say the viral nucleic acid is. We don't know for sure if it's infectious virus or not. And it goes out for a long period of time. We can track it in the animal out to 42 days in the lymphoid system as well as the small intestine. Remember that. I'll get back to that because that's contrary to what the actual tissue virus localization data is. All right. Oral fluids, are they a good diagnostic sample? Yes, they are. As soon as the pigs were old enough to start chewing on ropes, we started collecting samples. And yes, indeed, they are positive. All the different litters, except the contact control, or the negative litter, they stayed that way. But certainly the virus is being shed in those litters. You can see the ones that had 100% mortality. No samples there. 21 days, we're still picking it up. 28 days, we're not seeing as much, but we can still see some positives. It's, for all practical purposes, gone by 35 days post-inoculation. So we have about a 3-week window that we can actually use oral fluids and ropes to surveil for the presence of this virus. And the PCRs work very nice. The sow data, that's kind of really interesting. So no detectable viremia in the sows, but really that's not unexpected. We only saw it up through day 4 in the baby pigs. We did weekly collection on the sows, so it's flat negative, not a surprise. Fecal shedding, basically the patterns that we saw in the sows, or in the baby pigs, were reproduced in the sows. So the days that the baby pigs first showed was also when the sows started showing, even though the sows really didn't get all that sick. Virus loads were not nearly as high, but they were clearly detectable. Both fecal shedding as well as nasal shedding. And then at 35 days post-inoculation, when we sacrificed the sows, once again, we saw the trend for virus being present in lymph nodes and in some of the small intestines. That's 35 days post-inoculation. Now, is that infectious virus? We don't know at this point in time, but it's a clear PCR signal. So, potential is there. Virus lesions and immunohistochemistry, those are the main points that I'm going to talk about today, and the big reason for that is the Sarah at UNL is writing most of this up for her PhD thesis, so I don't want to steal her thunder. I'm just going to speak in generalities. Most of the lesions that we saw, histopath lesions, this is H&E sections, villus blunting, really extensive destruction of the villi in the small intestine. This slide right here is not an H&E section. That is immunohistochemistry. And notice the villi. Virtually every cell in the villi is just loaded with virus. That's why the diarrhea is so severe. So, I mean, that was really, really surprising. That's 1 day post-inoculation. Also, this is the time that the virus is leaking from these infected cells through the lamina propria into circulation. higher magnifications. Some interesting things, and I was talking about pathogenesis. We have immunohistopositive cells in the lamina propria here. What are those cells? How does that deal with the mechanisms of pathogenesis? These are going to be future studies to figure that out. Antibody response. We have initial SN data and immunofluorescence data on these pigs. They are virtually 100% seropositivity rate for both assays by 14 days post-inoculation. We're in the process of fine-tuning these assays, so I'm not going to give you titers and antibody response over time, but we see clear-cut immunoresponsiveness by 14 days. The systems are being worked out. The samples that we have collected, we're sharing with other laboratories across the U.S., so the whole benefit to the industry is that these samples will be used to standardize the assays that you're going to be relying on in all the different labs. Okay, special thanks. So I've already listed my collaborators that we did this work with, but I also want to give special recognition to Dr. Sabrina Swenson and Melinda Jenkins-Moore from the National Veterinary Service Laboratories. They're a selfless group. They share their viruses with us. Those are the viruses that we use to develop the SN and IFA test. I think it's important that all the labs share with the reagents that they generate over time. This is the way that we need to do business and not just hog it all. So I think that's where yes, I'm done. Questions, or how do you want to do that?
We'll maybe keep it at the end here.
Okay.
Keep kind of rolling on. All right. Ready to roll? Okay. It's good to see all of you here today. Really good the reception last night, and good to interact with you and stuff. You know, we interact with email and stuff with many of you all the time, but it's really good to see a lot of you face to face. Also, it's a little intimidating here. I mean, going after a world famous virologist like Dr. Hesse, I mean, he just told you Koch's postulates were fulfilled. For a virologist, that's like Mike Tokash with the Vikings, the North Dakota State and K-State all winning in the same year. You know, this is like he rung the triple threat here in his career. So he's you know, it's kind of pretty phenomenal what he's done there. And also, I got to take a hats off to our diagnostic labs. We travel around the world, and you would not believe how lucky we are for our diagnostic labs in the U.S. And Dick has been a key part of that. Roger Main at Iowa State, Jim Collins at Minnesota, the South Dakota crew. It's just been phenomenal. You think about this, last year at this time, we were just getting the first reports that this disease existed. They didn't have diagnostic tests online till in March. And all of a sudden, and then the pork board geared up with funding. So we've learned a lot, and it's phenomenal the amount that we're learning over time. So I'm going to kind of give you the background on how we got involved from the feed side, and Jason's going to give us an update on some of the research side. First off, many of you have seen this. If not, it's something we're watching. I just want to give a little caveat here. I'm cautiously optimistic, but by March or April, I'll be able to be a little more relaxed if things go the way that we hope they do. What this is an accounting of cases in sow farms in the U.S. Accounts for about 40% of the sow farms. Done by the University of Minnesota. They started this with PERS. Now they're doing it with PED and Delta Corona. And you can see we've had a fall off in cases, and we've stayed pretty flat line over the time period here. Where I have this black line is kind of where we drew a line in the sand and said we need to provide some options. We need to start doing something different from the feed standpoint. Now, This is over time, but it sure looks like we've had an influence on the number of cases. And so that's my objective here today, to give you a little more background on where we're coming from and where we're going. This was some of the data, thanks to the Pork Board being on their website. We started seeing this in January, and when we started, first off, when I saw the infectious dose study that they did, and they had to redo it because it wasn't dilute enough, or, you know, I won't get into all the details, but they had to do it twice because of it's way more infectious than they thought it was. And so basically, if you dilute baby pig feces, you know, you can dilute it in like 700 gallons, and it's all 700 gallons are infectious. Another way of saying this is you could infect about 3 million pigs with that 1 gram of acute baby pig diarrhea. So it doesn't take very much of that acute diarrhea to spread. And just to give you a little history, in May, you know, I'm lucky enough, I knew something serious was going on when Dr. Henry gives me a call one day and says, you need to review your biosecurity at the K-State swine farm. I mean, I knew that was an urgent situation. And thanks to our diagnostic lab, they upped their biosecurity. After that phone call, I knew this was going to be a big deal. And we started getting reports that, you know, when we found out this was Asian, and we get a lot of questions about the vitamin premixes and the products coming out of Asia. And, you know, Mike and I were involved in some of the initial surveys the USDA was doing, helping them with those questions. And we were struggling figuring out what's our role, you know, because we're hearing the devastation of these cases, and we're trying to figure out how can we help and what can we do. And in the initial part of this, we were kind of like, you know, I went from every veterinarian convincing us that we weren't feeding enough vitamin D to all of a sudden they're pulling all the vitamin, doing crazy crap, like pulling all the vitamins out of their sow diets. You know, it's like that is not the right way to go. And so we're just trying to figure out there. And we didn't think, knowing the way, looking at other diseases and all those kind of things, we didn't think feed would be a major transmission route. We thought it'd be a possibility, but we didn't think it'd be a major transmission route. And this was one of the initial trucking study. The amplification of the virus, this is a key thing that we've really learned in our industry. The amplification through packing plants, the understanding of that is a big deal. Packing plants are a nidus for infection and amplification. And once your packing plant gets infected, it just goes everywhere. And so you really got to control that critical control point. The other point is cull sow yards. This is another picture. Matt Turner, they knew in North Carolina their cull sow assembly yards were positive in June. And then it took a while in their industry to build up the virus, and it didn't blow up until in the fall. And we've seen this. Laura Greiner, they were able to see sow farms break due to a study they were doing. The virus was in those farms long before a few days before the clinical signs even occur, and it just goes epidemic. And you produce all this virus, and the whole system gets contaminated. And we'll come back to that, but it's critical. We just can't let this virus circulate throughout our system. Then, thanks to, we've got a whole network of people we interact with, you know, and veterinarians and stuff. And we started getting, as a group, a lot of questions, and we started getting some cases. And things in production systems that are doing a great job with diagnostics and records, having this common theme of clinical signs appearing in the early nursery, and we'd still have upstream farms that were negative. And we had this multiple cases. We have about 5 or 6 different cases where we could go through this same story. And so I picked one of these with a farm here in the Midwest that we've worked with a long time. It's about 50,000 sows. They got 18 sow farms. They had 2 PD positive farms, one in November, one in December. They went through, of course, once you had a break, and they made the decision. They focused on everything they could from a biosecurity standpoint. About early January, we made the decision we're going to remove all the creep feed out of the sow farms. And I think that's a critical thing in this case. Haven't had a sow farm break, knock on wood. I'm cautiously optimistic we'll know in the wintertime, but they have not had a break since. About 9% of their production system's positive versus over 50% of the U.S. industry. And this is as I said, we started getting these commercial nurseries that were going positive out of the negative sow farms. No clinical signs in the sow farms, but we would get diarrhea and excellent diagnostics in the nurseries, and those sow farms would continue to be negative, and they were continually going positive right after arrival. So was it trucking? Was it the truck wash? Was it stuff like that? The veterinarians started doing a lot of environmental sampling. We started doing some feed sampling and starting looking for PCR positives here. Then within their production system, they also have, this is the case that really brought it to the forefront. We happened to be there on a visit on site and started to review some of this stuff. And they've got a gilt multiplication nursery. This is a continuous flow nursery site that they've used for gilt multiplication over 25 years. It's never, either this multiplier farm or this nursery has never had a PERS infection introduced. And I say that to illustrate the biosecurity of this site. It's an isolated site. It's enhanced biosecurity, and very good. And then they move they'd wean twice a week into this nursery site from this sow farm, and then move once a week. They move out into multiple GDUs. They move 50-pound gilts out into those GDUs to feed the other sow farms. Well, this nursery had diarrhea noted in relatively newly weaned pigs, few days weaned. Feed sample. The service person had submitted a feed sample, came back PCR positive. Over the next few days, clinical signs of diarrhea continued to increase throughout the next 3 days throughout that site. Subsequent testing, again, came back with fecal swabs, came back positive. It was clearly PED. Clearly, clinical signs moved through that site from the younger to older pigs. And this is where we got really, really, really lucky and shows the value of good diagnostics, good diagnostic labs, good cooperation with the veterinarians, and good communication between us and knowing what's going on, is they had weaned into that site at least twice when they had clinical signs and did not transfer it back to that sow farm. Also, when they found out about clinical signs in that first week, they shut down all the movement into the GDUs. And they were able to contain the virus within this nursery site and not contaminate these GDUs. This was very, very, I'd call it lucky, but very good, very skillful from the veterinary support side. So, we recommended, again, I mentioned, we removed all the creep feed, nursery feed products from the sow farms because we were starting to get some of the data from Minnesota showing the ability of the virus to be in feed products, and we're concerned about some of the porcine protein products. In February, we removed all the porcine, we changed the diets. We just drew the line in the sand, said, we've got to make a change in this system. Talked it over with the management. And this is where it really became interesting. Here, this is what this is the commercial nurseries that were being filled. 12 of 21 of them were positive with clinical signs. In January, February, we had this string of positives, and the sow farms had stayed negative into these nurseries, into these commercial nurseries. We made the diet change, and it was just like a light switch went out. Somehow we had inhibited transmission. As I mentioned, in this production system, no new sow farms to date. As of October 31st, we had only had one unknown infection in a nursery. Just talked to the veterinarian last night again. Again, cautiously optimistic. We've made it through the manure season. Nurseries continue to seem to do well. I'll be a lot more optimistic in March, I'll tell you that. The other case that I want to share with you is, I'm sure you know some of the details of this one, is at the same time, got to go out, the Canadian veterinarians and Canadian industry itself, very concerned about transferring PED back from the U.S. There's a lot of pigs, a lot of trucks, things going back and forth. They put a lot of effort into monitoring and getting an early warning system in, and they thought their first cases were going to be in finishing barns, you know, drug in from trucks back from the U.S. All indications were it was going to be that. Well, in January 22nd, they had their first case, and it was in a sow farm. Then over the next week or two, they started having other cases, and they've got a really good epidemiology group at the University of Guelph, and they made the observation that 10 out of the 11 of those first cases were associated with the same feed company. So they started testing the transport vehicles and trying to review all those product protocols. Couldn't come up with anything. Again, these farms are all geographically diverse. They're farrowing in nursery, much different distribution than they were expecting epidemiologically. Something wasn't adding up. And then within this company, they had two lines of starter feeds. One that was porcine protein free, one that was with plasma, blood cells, porcine proteins. In all 10 of the cases associated with this company, were being fed these plasma-based diets. So epidemiologically, it's becoming very, very strong evidence of something's connected to the ingredient or with that diet. This company did a pretty dramatic thing. They did a voluntary recall, recalled all the production made with once they got confirmatory evidence that the plasma was PCR positive. They did a company-wide recall voluntarily. Probably a controversial thing at the time. And then they submitted subsequent testing. The Canadian Infectious Disease Lab in Winnipeg, probably one of the premier infectious disease labs in the world, came in, did bioassay on the plasma. It was clearly positive. Bioassay in the feed was not positive. And so that's led to some confusion here. How you can have a positive ingredient but not have a positive assay in the feed. And so led to a little difference of opinion. But we got to remember that the sensitivity of bioassay is you're putting a small amount of feed into 3, 4, 5, 6 pigs. The sensitivity of that bioassay is not very high, and so when you get a positive, you know it's a positive, but when you have a negative, you don't really rule it out. And then epidemiologically, there's another piece of evidence in this case. There's a little place called Prince Edward Island over here. It's on my bucket list to go visit someday because it's like supposed to be this beautiful place. And there's 20 pig farms on the whole island, and like half of them are gilt multiplier farms, high biosecurity because it's on an island, it's isolated, there's not many pigs there, and they and high biosecurity. They were the second province in Canada to get a PED case. And when you looked at it, this was about 4 days after this company did the recall, and when they started looking at, they had fed pellets from the same company in Ontario, and it was the plasma-containing pellets. So epidemiologically, that was about as close to causation as you could get. And this was before all the, we had any of the bioassay and those kind of things. So got to give the Canadians credit. Again, they're cautiously optimistic. Most of the sites, they've limited to about 69 cases. They did have a case last week again, and they're cautiously optimistic that they can keep this out of their whole country and clean up their country, and we're hoping that they do from that perspective to really show us how things can work. Again, just briefly here, showing the distribution of cases. We're in farrowing and nurseries, and they thought that it was going to be finishing barns, and you can see how after the feed recall, it stopped the transmission here, and then we've got many more secondary cases coming on that are coming through the finishing, coming through the transport side of things. So, I'm going to turn it over to Jason. I'll be back with you just for briefly here, and Jason's going to give us an update on the path we've taken down the research road. He gets to talk about some real data.
That's the nice thing about it, though, Steve. But, on the other hand, though, you said you're intimidated following up Dr. Hesse. Well, I'm the nutritionist talking in the PED session, following two veterinarians as well, and so we'll kind of see where this goes. But as Steve said, he did a really nice job setting the stage for the connection between PED and feed. And I'm going to follow up then on some of the research that we've been conducting here at K-State to also explore this particular area. And before I get started, kind of similar to what Dr. Hesse said, there was a giant team of experts and people and from all over that really came together to get some of this research conducted. And I'd first like to start out by thanking the group there at the K-State Applied Swine Nutrition team. Dr. Srimas Nidakanshana was a past PhD student that we had that did some of the original literature review that led to some of the research that we'll be doing today. Hopefully you'll get a chance today to talk to Lonnie Schumacher. She's a veterinarian that's just recently joined our swine nutrition team. I'm happy because I'll be passing on this PED torch to her to carry as we go on further, and so she's done a super job thus far. Then the rest of the team here, Dr. Streets, myself, Tokash, Drew, Good Band, all did a really good job supporting the work. Worked a lot with the grain science department here with Dr. Cassie Jones and some of the work that she's been doing. I'll be talking about some of her research in my third study I'll cover, but also Stark, Dr. Huss, Roger Cochran. I'll be using the word we a lot during the presentation today, but that we is really code for Roger Cochran because he was the one that did a lot of the work through a number of these studies right here. The K-State Vet Diagnostic Lab has been a tremendous support. Dr. Hesse, Dr. Jennifer Bai, Dr. Huss, Anderson, and the rest of their team, as well as Steve also already mentioned Iowa State Vet Diagnostic Lab, a tremendous group of people up there. You might recognize the name Roger Main right here, a past product of the K-State Swine Nutrition Program, who's taken the charge there at Iowa State to lead that vet diagnostic group. His group there with J.Q. Zhang and Phil Gogger and the rest of their team have done a super job. And then finally, none of this could have been completed without the support and the foresight of the National Pork Board to come together and say, you know what, we've got to do something to figure out how to really mitigate this PED problem. So a lot of gratitude given to the National Pork Board and what they've done to help further our knowledge of PED. So what I'm going to be doing today is talking about 3 different studies that we've conducted here at Kansas State University with PED and feed. The first study will be a study reviewing some work we did to establish the minimum infectious dose in feed. Steve already made or alluded to the comment that it takes a very little amount of PED in feed to actually make pigs sick, but ultimately we didn't really know how little that little was, and so we ultimately conducted some research to really pinpoint that. Followed that up then with some additional research looking at thermal processing. Can we use a typical conditioner pellet mill setup that everybody has out there in their feed mill to help mitigate the risk of PED? Then following up with some of the post-manufactured chemical mitigation work we had. Again, what's really interesting, really unique about this chemical mitigation work that we have is it has the potential to allow for some residual benefits past just a simple point-in-time kill step. And so definitely all of this, again, work conducted by or supported by the National Port Board, and we thank for that. So let's get in right now, talking to some about the data. The minimum infectious dose study we conducted, we started with a concentrated dilution of PED or solution of PED. Did a serial 10-fold dilutions with this mixture to form 10 decreasing concentrations of PED. Those solutions were then mixed with 4.5 kilos of feed, blended, as you can see down here, in a lab scale pellet or a paddle mixer for about 2.5 minutes to make sure and homogenize and distribute that PED virus. The blended feed was then added to PBS and sat overnight, which is what you see in this picture here. It was that solution that we pulled off of that mixture with the PBS is what we actually gave to the pigs during our bioassay. And so ultimately the feed samples here were analyzed both for PCR to look at the presence of PED RNA, as well as put into pigs to look at bioassay to really confirm infectivity. So to look at some of the results right here, let's take a minute just to set up the slide. What you see along the horizontal axis right here is the concentrations of the virus in both tissue culture media, which is the blue bars, and then the feed, which is in the red. The highest concentration of PED is on the left axis, going gradually over to the right in a more dilute fashion. In our vertical axis right here is the cycle threshold, cycle times, CT counts, and we'll be referring to that a lot. And it's important to remember that a higher CT means less PED RNA was detected in the sample. And so our goal is to have as high of a CT as possible. Anything low means that there's more PED RNA present in the sample. And so let's take a minute and look just at the blue bars right here. This would be the results of the PCR analysis in the tissue culture media. It was this tissue culture media that we added into the feed to inoculate the feed. And you can see that as we reduce in 1 log increments the concentration of PED, we see about a 3 to a 4 CT count increase via PCR analysis, which again confirms that we have less virus in that tissue culture media. And it's nice that as we reduce again by in 1 log increments, we have a fairly consistent reduction in the amount of PED RNA that was detected. The red bars then are the PED that was analyzed for in the feed matrix. And so we put this tissue culture media into a feed matrix, then analyze that feed. You see, first off, a significant increase in the amount of PED that was detected via the PCR analysis, about a 10 CT count increase in PED right here. And so, again, what that would suggest is that when we put this PED into feed, there may be something in this feed matrix which would be beneficial for us because we have less PED RNA that's being detected. You know, the big question, though, is that just because it's not being detected in the PCR analysis, does that mean that it's not infective? So ultimately, we have a good indication, because we end up following this into pig, that at least the positive PCR analysis were associated with infectivity in the pigs. But I mean, there could be something in that feed matrix that's binding to the PED, allowing it not to be detected via the PCR analysis. Could be a simple desiccant effect, destroying the PED. Maybe something else in that feed matrix is also acting to destroy the PED. And so ultimately, what we found is that the 4 highest concentrations of PED that we placed in feed were associated with positive PED analysis in the feed. One thing that's also important to remember is that for this particular analysis right here, we used a threshold of a CT of 45 to determine the difference between positive and negative. And so anything higher than 45 was deemed negative. Only the 4 highest concentrations right here in the feed were positive. Again, I want to point out that this lowest dose that was a positive sample had a CT range of 37. I'll come back to that here in a minute or two, and we'll kind of comment on that. But as we go to the next slide, this shows the results after we fed this inoculated feed to these pigs. And so just to set this slide up again, this would be the 5 highest doses of PED in the feed that we measured, the highest dose being at the bottom. Then across the columns right here, we have the day that they were inoculated, and then 2 days post-inoculation, all the way up to 7 days post-inoculation. And this would be fecal swabs, and then the PCR analysis of the fecal swabs. And so to make it simple, the blue box right here would indicate all the positive PCR analysis from the fecal swabs of these pigs. Ultimately what this means, and Dr. Hesse showed some really nice pictures up here. I don't have anything like that here to ruin your appetite, but basically you can imagine the same thing was observed in these pigs too. So we had positive fecal swabs right here when we fed positive feed to these pigs. The big thing right here, and what we definitely determined, was that this dose of 5.6 times 10 to the first is an extremely, extremely low dose. Was able to create pigs being sick, again with a CT, if I go back to the last slide, of about 37. What's really, really important to consider right here is that early on, some of the diagnostic labs were using a CT value of 35 to determine whether a sample was positive or negative. So anything above 35 would have been deemed negative. This data right here would indicate that if a CT of 37 can actually make pigs sick, there's a potential that negative samples can still lead to infectivity. So it's one of the things that you need to think about when you start sending in samples and looking at for PCR analysis is where their cutoff for that lab is and what they deem as something to be positive versus negative. Right now, I believe a lot of the labs are going to a 40 or a 45 cutoff range also, and so there's a little bit more cushion right there, but definitely important to understand what these PCRs mean, and a negative may not necessarily mean that it won't lead to infectivity. Ultimately, what does this study really mean to you and I here in the field? It takes a really low amount of virus to be able to infect pigs. Roughly 200 infectious particles in the feed been associated with infectivity. To put this into comparison, one acutely infective pig will produce about 10 million infectious particles. So definitely, again, we keep on preaching it, but the importance of biosecurity to prevent tracking very small minutes of manure around to infect pigs. So when we look back at our study, our minimum infectious dose is the equivalent of putting 1 gram of feces in 500 tons of feed. And if there's just that little amount of feces in that feed, that's enough to actually make the pig sick. So again, emphasizing biosecurity, and we can't say it enough, really the importance of ensuring that we're not accidentally tracking this material around as we're going from farm to farm or into the feed mill. You know, it's one thing that Steve mentioned a little bit, and I know we've had a lot of other discussions about just how this virus can be getting into the feed mill. When you're only talking about 1 gram in 500 tons, it doesn't take that much. And so really important research right here that we can then be able to utilize as we go forward in some of the other studies. Which leads me into the second study. Some of the work that Dr. Nitya did show that there's a time and temperature relationship to the destruction of other coronaviruses. And the really interesting thing about that is that potential kill curve also corresponded to a range of time and temperature that would normally be observed in the normal pelleting process. And so we formulated this study to look at a low dose and a high dose of PED. Then had 3 different conditioning times, from 45 seconds being a relatively short retention time, 90 up to a very long retention time at 180 seconds, and then conditioning temperatures of 155 degrees, which is relatively low, to 195, which is really, really high. Again, tested these samples for PCR analysis as well as fed them to pigs for the bioassay. Really interesting opportunity we had to be able to work with this piece of equipment. This is a smaller benchtop size pellet mill that we were able to utilize. Thanks a lot to Charles Stark for organizing this. You know, this work wouldn't have been able to be done if it wasn't for this piece of equipment right here. And the scary thing is, for us to do this work, we needed to create 6 liters of PED virus to be able to inoculate enough of this feed. If we had to go to a larger scale pellet mill, we would never have been able to grow enough virus to be able to get these results right here. And as I mentioned before, the we is here dumping the feed. There's Roger dumping that feed into the pellet mill. So you know who does the work around here. These are the results from the PCR analysis of the feed before and after it was processed. And so on the left-hand side is the low-dose PED. On the right-hand side is the high-dose PED. And so let's start at the bottom, the low dose right here. The tissue culture had a PCR, or had a CT of 20 during PCR analysis. Once we put that in the feed, the resulting feed had a PCR CT of 31. Again, corresponding to about a 10 CT increase when we put that virus into feed. However, it didn't matter that much from a relation of either different times or different temperatures. We ended up seeing about a 6 to a 7 CT increase on each of these samples when we put it in through the increase in temperatures and the increase in time. Very similar pattern we saw with the high-dose PCR, where again we had a more concentrated tissue culture media, which again resulted in a higher concentration of PED in the feed, about 10-fold increase. Then again, another 6 to 7 CT increase when we put that through the pellet mill, temperature and time combination. So the next question then comes to the bioassay. And so we've conducted the bioassay already for the low dose. The high dose is actually on day 5 today. And so there's pigs up at Iowa State right now that are consuming this feed or had consumed some of this feed. What we've seen thus far is that when we fed these feed treatments to the pigs, the negative control, which was the unprocessed feed, we're able to induce infectivity. Pigs did get sick right there. However, all of the treatments right here did not result into any infectivity of the pigs. And so this would be some of the first work that is available out there that might indicate that we do have a piece of equipment in everybody's feed mill right now that could be available to disrupt infectivity via PED. The important thing, though, to remember is that when you start thinking about a pellet mill, it's a point-in-time kill step. So if there's post-processing contamination, you won't have any benefit at that point. And so that's what really brings us in to the next study that we conducted. It's a study, again, that Dr. Cassie Jones and Roger took the lead on here. It was a sister project of another study that was funded by the Fats and Protein Research Foundation looking at salmonella. So again, there'll be some other salmonella-related work very similar to what we had right here. We started with 4 different feed or ingredients that we treated. Looked at a complete nursery diet, looked at a porcine meat and bone meal, a blood meal, and an animal plasma. And then we treated these 4 feed matrices or ingredients with 7 different chemical treatments. We had a mixture of organic acids, a mixture of essential oils, medium chain fatty acids, sodium bisulfate, commercial formaldehyde, which is a terminate product, you'll see it referred to as, and then sodium chlorate, and then we had an untreated control as well. Just to give you a little bit of idea of what we did, we secured all the different feeds in the feed matrices, deemed them to be negative for PED via PCR analysis. Then we applied the 6 chemical treatments and the untreated control. To those treatments, we then inoculated the PED. And so we first treated the batches with the chemical treatment and then inoculated with the PED, so showing a post-treatment contamination of the feed with the PED analysis. Then had 3 replications on here, ended up storing it for a period of 42 days, conducting PCR analysis over that 42-day period. This study was only a PCR-based research, and so there wasn't any bioassay associated with this one. If we start looking at the data slides, I'll have 4 slides that look just like this. I want to set those up. First off, up in the title, you'll notice right here, so this is the swine diet slide. You'll see another one for the meat and bone meal for the plasma as well. Along the horizontal axis is the days post-inoculation that we collected samples from day 0 when the inoculation occurred all the way out to day 42. And then again, the CT values along the vertical axis. Again, a higher CT value is what we're looking for right here. And so first off, when we start looking at these slides, we should take a minute to look at the black line, which is right here, which is the untreated control. So when you look at the pattern from day 0 to day 42, you'll show the normal deterioration of the PED just over time. So just sitting this sample at room temperature had a certain impact to increase the CT in a swine diet right here. Then looking at the different lines associated with all the different chemical treatments right here, you'll see some changes, a little bit of a bump up here with the essential oil, but really the one that stuck out was the medium chain fatty acid treatment right here. It's something that when we started doing a literature review, Roger dug in and found a little bit of information, and we put it in here with our fingers crossed, hoping something would work. But definitely it looks like right here, and as we start talking to more people, there maybe is some other science and some other viruses out there that could, and other pathogens that could help support the belief that this medium chain fatty acids actually does have an impact on mitigating PED, at least from analysis via PCR analysis. Looking at the blood meal, again looking at the black line right here, you'll see a certain increase over time in PED presence, as at least detected via PCR, showing a deterioration or a less RNA being detected. Again, some bouncing around with some of the treatments, but the one that really sticks out is the essential oil product right here. So you see after day 7 to day 14, a significant increase in the PED PCR analysis right here, an increase in the CTs showing less RNA being detected with the essential oils in this blood meal. But it's interesting then when you start looking at the meat and bone meal right here. The first thing that you'll see is, these slides were all on the same scale, but look at how low the PCR analysis was for the meat and bone meal. You'll also notice that if you look at that black line, there's very little change from day 0 to day 42 for the untreated control. And so it appears, number one, that there does not appear to be any deterioration of the PED RNA over time in this meat and bone meal. You'll also notice right here that it did not appear that any of the chemical treatments had a significant impact on increase in the PED PCR analysis. When you switch then to the animal plasma, which Steve mentioned a little bit too, again, look at the black line. Very flat from day 0 to 42 change in CT, again indicating a certain stability in the PED in this blood or in the animal plasma, at least detected via PCR analysis. Other than the treatment with the terminate, which is a formaldehyde product, everything else seemed to have a certain or not much impact on the PED CT levels right here. This impact or this observation with the terminate of the formaldehyde would also be similar to some work that Dr. Scott Deed did up in the Pipestone veterinary system too, where he also noticed the formaldehyde potential benefits on PCR. This slide shows the 4 feed ingredients in the complete feed, and just looking at the untreated controls from day 0 to 42. And again, we threw this up there just again to show the normal deterioration of the virus over time. And again, you see in the blood meal and the swine diet a certain increase that would suggest less PED RNA being present, at least by day 42. However, there's something that's going on here with this porcine meat and bone meal and this animal plasma that leads to a certain stability of the PED virus in those 2 matrices. And so definitely it looks like when we start talking about PED, we also need to be mindful that different matrices, different ingredients may have a certain impact on whether that PED will be stable, whether it be present, and be able to be analyzed for. To kind of close it up, what have we learned here with the K-State studies that we've done with PED? Number one, we lose about 10 CT counts as we put this virus into feed, which is a good thing. I mean, hopefully, and balancing back with my minimum infectious dose study, it looks like when we do put that virus into the feed, there's something in that feed matrix that increases the CT that potentially could result in less infectivity. We did confirm again that a detectable CT can result in infectivity, which is important to continue to remember. And again, the minimum infectious dose of PED can be higher than the cutoff of some labs that are utilized out there. One of the nice things, and again from the science, from the research standpoint, that we can detect low amounts of PED on a very continuous and repeatable basis, which really sets the stage for a good research platform that we can continue working on this particular area in the future. We've noticed that there are potentially some opportunities for chemical mitigants to act to minimize PED contamination. Definitely, though, it looks like it's matrix specific. For instance, the formaldehyde in the plasma or the medium chain fatty acids in the feed look to really have some benefits that definitely warrant further investigation. And again, finally, don't want to beat this too much, but it sure does appear that there's something with plasma and meat and bone meal that leads to a certain PED stability, and something we need to be aware of when we're formulating diets going forward. So again, the only thing I'd really throw out to caution right here, at least from the last study, it was all PCR-based analysis. There wasn't any bioassays conducted on that, and so we have an idea of how much RNA was detected, but do not have a clear idea of how much infectivity was associated with those particular PCR analysis. So with that, I'll hand it back to Steve and let him close up this session and take us home. So.
All right. Thanks, Jason. Good job. It's great to see we've been able to generate some data there. Just going to give you some quick, just give you the 3-minute summary here. This is an area we've been working on, focusing on. You know, it came last March. We have this big detailed biosecurity plan for our farm, and Charles Stark asked me, what's our biosecurity plan for our feed mill? And I was like, ooh, man, that's a big hole. So anyway, we've got one now, and we've been working on this. So don't know what happened there, but there we go, wrong way. Big thing, as I mentioned, ingredients in the feed mill. Breeding stock, boar multiplication diets, we've pulled them out of mills. We're going, at least until we get this thing figured out a little better, We prefer using mills with no porcine proteins for the breeding herd. We've had a lot of people, a lot of feed mills and stuff. They've done an excellent job of helping us with sequencing. It becomes complicated sequencing through a feed mill because, as Mr. Cast had pointed out, you got to sequence not only production, you got to sequence the loadout, you got to sequence the truck. There's a lot of stuff going on there. Ingredient receiving. I got a whole presentation of the things we've been seeing at ingredient receiving. Just remember that slide adjacent, how much poop it takes to contaminate feed. And if you walk across the grate with poop on your boots, you're potentially infecting your sow herd. If trucks are coming across those grates, we got a lot to do at ingredient receiving. Believe me. I know when you got 50, 60 semis lined up, farmers do not want to wait. And so, but we got to change things at ingredient receiving. Transport protocols, a biosecurity pyramid for feed delivery. Jason mentioned Scott D's published an article. Right in there it says this sow farm broke, connected with, they had an emergency feed order. They'd had a biosecurity transport pyramid. They broke it with an emergency feed order and led to 3 sow farm infections. Out on the finishing side, we need to know the status of sites so we can deliver to those positive sites. Assessment of the feed mill as part of biosecurity. Just like I said, we hadn't even thought about it here at K-State. And so we put together a plan. PIC's put a lot of resources into, as a company, developing a biosecurity plan for their feed mills. Okay, as we wrap it up here, just want to go through some of the things we've learned from the coronavirus biology. Just remember, this is a virus. It doesn't multiply in the feed. We're lucky on that step. So if we can do anything on a kill step, we can reduce infectivity. So anything we can do to prevent recontamination, those kind of things, will be a positive thing. Remember, the infectivity of this is a big deal. Newly infected farms, too. Remember, Dr. Hesse showed that graph of shedding. And Dr. Hesse and Henry last year showed us in a baby pig, you know, an individual pig excretes a tremendous amount of virus, but it's only for a little amount of time because it just destroys these cells and then it destroys the pig, and they're not around to shed for that long. So we got to remember the limited times of periods. And remember that we put a lot of biosecurity into going in and out of buildings. The environments around these farms, there's so much virus in the environment that it isn't just the building itself, it's the ground around outside, and especially in the wintertime. You got the large volume of dead pigs. Dr. Hesse showed the famous picture that I hate to even think about. Nursery and growing finishing sites. This is something where the attitude's got to change out there. The attitude is, I hear it in a lot of production systems, it's not that big a deal, it doesn't impact growth, it's not that big a deal, it doesn't kill that many pigs. Mild clinical signs. The problem with nursery and growing finishing sites getting infected, you start amplifying this virus through your system. It amplifies to the packing plants, it amplifies to the truck washes, it amplifies throughout the whole system. So it is a big deal out here in grow finish to control the infections, and you gotta remember that a lot of times if you aren't doing a lot of diagnostics, you got to treat these sites as positive. On a better note, on a positive side, working with production systems, basically, definitely something is going on out there. The better biosecurity systems, the people that are doing a lot of things right have had a lot lower incidence of infection. So we know these biosecurity procedures work. You just got to apply them and have the discipline to get it done every day, like a lot of lot of you do, and a lot that I know you do in your production system. Been able to, a key thing, been able to segregate out that trucking, washing and drying trucks. You know, we've talked about it for years with PERS and those kind of things. Managing these risk ingredients in the mills. We eliminated creep feeding. We've put that on hold for now. We did the research. You know, Ramel, he convinced us to creep feed and the importance of it, but we've kind of put that on hold for now till we figure this thing out on the sow herd side, because that sow herd is at risk. Eliminating the porcine proteins from diets. The critical thing, I was talking with Dale Kiesucker earlier here. Luckily, we've had this great group of grad students that have done a bunch of amino acid work, and we've learned how to feed pigs. And you know what? We can actually feed them cheaper without plasma protein in the diet. We lose a little growth, but you know, the emerging evidence out in the field is it isn't that big a deal. And I can't emphasize it enough, don't let this virus become extreme within your system. You know, and we've got many examples of farms being able to clean up as well. Began producing negative wean pigs. The PIC system, they've cleaned it up in a lot of their systems. And I think we really got to take our hats off to the Canadian industry. We've got a lot to learn from them, from their cooperative ability within their industry as well. So